Everyone comes in, welcome so much to the Global Virus Network's Forefront of Virology webinar series being hosted today in our short course. I'm Maggie Bartlett. I'm the program director for the GVN, and I am so pleased that we have Dr. Sten Vermund with us. He is the Dean of the College of Public Health at University of South Florida, the Chief Medical Officer for the GVN, and is an internationally renowned pediatrician and epidemiologist who has spent a tremendous amount of time working on HIV and AIDS, primarily in adolescents and maternal health. And I believe in 1993 changed the case definition for how we think about AIDS for the CDC. But I don't want to embarrass him. I'm just very glad that we're going to get to hear from him today. So without further ado, I'm going to hand it off to Dr. Vermund to give us an overview on translational virology.
All right, thank you very much. Those of you who are online will perhaps see my back as much as you'll see the front. We do have our short course in translational virology that just started today, and we have folks from all over the world who've come to Tampa, Florida for this course. And since it overlapped with the timing of the webinar, we integrated it into the course. So you see the crowd in front of you. So let's go to the first slide, next slide. I have a couple of disclosures. I worked for Moderna to teach them a course 2 years ago. I'm on a couple of scientific boards of startup companies that don't have any products. Next slide. So we'll talk a little bit about an introduction to translational research. Next slide. These are actually not the slides I sent. These are last year's slides. I've made quite a lot of updates, so I would like to use the slides that I sent, if that's all right. So as we get the proper slides up, this cartoon says the latest research says that we should do something with this research. And that's really the point of the entire hour, which is that we don't do as well with the bridge of discovery to application as we should. Zoe Morris and colleagues about 10 years ago did a now famous article where they took a number of examples and they averaged 17 years between the time of discovery and product development to actual deployment on behalf of human beings. And that 17-year gap is essentially deemed, thank you, is essentially deemed unacceptable. So let's go to the next slide. Thank you. The cartoon on the left, the cartoon on the right is more autobiographical. Shoot, we better hurry up and figure this out. These are aging researchers. Just kidding. Next slide. Not. Okay, and this is the slide from Morris, and Larry Green also did something very interesting around that time. So I give you those two references. And you know, you set priorities for research funding, you apply, you publish, you synthesize guidance and practice, and it's just too long. We also have the Valley of Death, famous in the field of biotechnology, where there may be a point where you have a product, it's promising, but nobody wants to invest in it. So it can be several years before perhaps it passes over that Valley of Death, and perhaps it's dropped altogether by a company. Next slide. I seem to be blocking you on this side here. So here are some historical examples of slow and fast implementation. When Edward Jenner in the late 18th century made his observations with cowpox as protecting little 8-year-old James Phipps from smallpox, and Lady Montagu's advocacy, you know, was also something of note. She was advocating for variolation and ultimately using variola minor to protect against variola major. But these concepts of cross-reacting immunity that they didn't understand in the 18th century, took about 50 years before European governments and North America used this as a routine. Benjamin Rush, who's a physician from Pennsylvania, who was a signatory of the Declaration of Independence in the United States, was very involved with this. So it was also known in the US. But it took 50 years before we started using cowpox to vaccinate against smallpox. Penicillin took 15 years, and you'd expect, what, penicillin? But, you know, from Fleming's original discovery, moving to Chain and Florey, we didn't have product until the late 1930s, and wasn't really available until the early 1940s. Then we have, and he discovered it in the 1920s. And then insulin was an exception. So people dying of diabetes who using animal insulin where essentially their lives were saved was so dramatic, and it was so easy to harvest insulin from animal sources that was a 6-month turnaround between discovery to practice. So we know we can do it. I don't want to dwell on it, but the COVID-19 vaccine, the SARS-CoV-2 vaccine, was the product of 20 years of development. When you look at what Carrico and Weissman did 20 years before it was published, and then all of the discoveries around nesting the mRNA within a lipid protecting coat, all of this was being done for 20 years. Just as COVID was released, we were prepared to develop an mRNA vaccine, and because we made the investment, Project Warp Speed, it was called. We were able to accelerate the trials. They still had 30,000 people in them, but we were able to mobilize them and fund them and complete them very quickly. And so we had the life-saving COVID-19 vaccine. Next slide. So here's kind of a paradigm for you that I borrowed from a Harvard website. You have pre-intervention work, and this is really basic science. Then you have efficacy studies, sort of at the bedside, clinical science. Then you have effectiveness studies, bringing the product into widespread use. Then you have implementation science, which is bringing a product to nationwide use, to routine use, to global use. And these are all different constructs, and we tend to have different scientists engaged, and all of them have translation elements. T1, T2, T3 are three phrases, terms you'll see, translational ones. So there will be basic scientists who are not focused on how the discoveries will be applied. They're just trying to figure out what this enzyme does. And they're not worried about how it's going to be applied. They're looking for fundamental discovery. But then there are other scientists who are very much focused on how to take a discovery that they made or somebody else made and figure out how it might apply to a product of some sort. It could be a diagnostic, a vaccine, immunotherapy, a drug, whatever it is. And then there are people like me who may come in and say, aha, this, lenacapavir works with twice-a-year dosing. Can we actually transform global AIDS control by letting people get a shot twice a year instead of taking a pill every day? And is that feasible? And we might do that in one area of one country. Let's take KwaZulu-Natal and see if it works. But then we say, aha, it does work. But we have costs, and we have logistics, and we have to be able to do, and how do we ramp this up to South Africa wide or globally? And then there's another level of implementation science. Next slide. And this kind of maps to basic science, clinical, public health, and implementation. Next slide. Now, this is another construct. I found this from Virginia Commonwealth University in Richmond, and I thought it was fun because it was simple. So this is what is translational research? Basic research, moving into improved or novel treatments and interventions, moving into community application. Next slide. So in microbiology, what do we mean by translational research? Well, laboratory and animal research that is focused on bringing a product to humans eventually. That is the purpose of that research. That's a form of translational research. Clinic to community, what our clinicians might do in partnership with public health or health services experts. And then community clinic to scale for impact, what we tried to do with PEPFAR, for example, the U.S. President's Emergency Plan for AIDS Relief, which was the largest investment in global health history, has now spent well over $100 billion and counting, and to try to reverse the catastrophe of AIDS in especially in Africa. Now the Brazilians were ahead of us. The Brazilians did this almost, it was about 8 or 9 years before we started PEPFAR. The Brazilians made universal diagnostics and treatment available to their own country. They did it on their own dime, which is worthy of mention. Next slide. Somebody's letting our guests in here because it's okay. Good.
You don't have to worry about it.
I don't have to worry about that. Okay. Next, we lost the slides here. I probably touched a screen or some horrible. Okay, I started my career very much as a clinical epidemiologist. You know, physician, studied public health, epidemiology. A lot of the questions I asked were kind of etiological. Does HIV cause an immune perturbation that will increase women's risk of cervical cancer? That was my PhD. And back in the 80s. And it was an important question because if it didn't increase risk, then it's one less thing to worry about. But if it did increase risk, we didn't want to save a woman's life using antiretroviral therapy, and then the woman dies of cervical cancer. And the hypothesis came from reading about cervical cancer and the fact that other causes of immunosuppression were a conspicuous risk factor for cervical cancer. So I said, well, I wonder if HIV is yet another reason for immunosuppression that might. And when we did the study and found out how dramatically it did increase risk, that had an immediate translational impact because every HIV program in the world should be screening for cervical cancer. And when you set up those sorts of programs, by the way, you should screen all women for cervical cancer. So that was sort of my early career.
We're back.
Yeah, so you can see why I'm interested in translational research. So when is basic research ready for translation? When you have cell culture guidance or appropriate animal model testing that show promise of a particular approach. When you have efficacy and safety demonstrated in these sorts of models before it gets to humans. But there are no clear established guidelines of when virology research is ready to go to humans. That is more like Delphic modeling. The Oracle of Delphi would bring intelligent people together and they would discuss things and make a decision. So we call that Delphic decision making. Next slide. So what factors are needed to design? You do need appropriate cell culture models, appropriate animal models. So often we find that an animal model might work from a virologic point of view, but it doesn't translate well to humans. All disciplines have that challenge. Do you know what animal model is the best predictor of human response for influenza, for example? Who knows? Maggie knows, she's not allowed to say. Ferrets. Ferrets. Now, who knew? This is what scientists discovered. That ferrets have a what about cardiovascular disease? Do you know which mammal has one of the best models for cardiovascular disease? Pigs. Exactly. Who knew? So you've got to find the right model. In HIV, for example, if you're talking about disease, you're going to do better with an SIV model in say, macaques. But if you're talking about infection, you might do better with an HIV model in chimps. And of course, we don't like doing chimp research because of the ethical concerns. So all sorts of challenges. Next slide. next bullet. Assess advantages of a drug, an immunotherapeutic, a vaccine, or other biologic of interest. If you can translate what you're doing in the lab and you can figure out, is it a diagnostic that you're developing, a drug, the world of immunotherapy, which is so exciting now, a vaccine, etc. Next slide. Next bullet.
I know what you mean.
So you know what I mean. Document the mechanisms of action with assessment of potential side effects and interactions. If you can do this in your cell culture first, your animal model second, you can radically accelerate human studies, because having superb animal data, say safety, efficacy, then we have a much stronger argument to be made for a phase 1 clinical trial in a small number of humans to see if it works. Next. And next. And then we collect data with FDA guidance. Now, there are many FDAs around the world. Food and Drug Administration in the US, the European Medications Association. There's a comparable group in the UK. After Brexit, there's a comparable group in South Africa. There's a comparable group in China. So all around the world. But to be honest, a lot of countries don't have the money to set up their own independent FDA. Let's take Suriname. They will look towards whatever the Dutch are doing, and and so the FDA is often used as data for countries to consider and other groups. Next slide. Now, why is it elusive sometimes to be able to move virologic discovery into translation? Sometimes we don't have suitable cell cultures and animal models. Next slide. Next. There are no NIH criteria for translational work in grants. Next. There's a gap in the physician scientist workforce. It used to be that most NIH grantees 50 years ago were doctors doing science. Today, those are a small minority. So you have to hunt for your clinical colleague if you're a basic scientist to find somebody who has an interest and motivation to work with you on bringing a product into humans. Next slide. And there is a failure of PhD and MD scientists to engage one another. We tend to live in our silos or our bubbles. And this is something that Clinical Translational Research Awards, CTSAs, Science Awards in the US tried to overcome. And many universities have these CTSA awards from the NIH, which are explicitly designed to bring the basic and clinical scientists. At Yale, when I was there, we had an HIV-oriented monthly seminar. And when we had a clinical topic, certain people came. And when we had a basic science topic, other people came. And we weren't achieving our goal. So we changed the structure and we did half an hour each. And guess what? Everybody came. Half an hour of hearing something that you don't do for a living, but it's interesting, is acceptable. The whole hour, you might go to lunch or something else. So we have to continuously work on this. Next slide. Then those are obstacles, but then how to overcome them, get the right model, ask the right questions, promote the best lab and practice scientists. Centers can often, overcome this. We think of departments as your topic area of expertise, but centers can cross the methodology and get people talking to each other. We have the K23 awards. Those of you from working in and from low and middle income countries can apply to the NIH for a career development award. It's one of the only career development awards I'm aware of that international people can apply for. And I've had amazing success of colleagues of mine in Peru and Zambia and South Africa and elsewhere who have gotten these awards from the NIH. And then we can nurture interactive meetings, seminars, and projects to get people talking to each other. Next slide. So something I'm going to talk about in a moment is my own experience in doing a clinical study. It might be a clinical trial, or it might be a sort of a clinical evaluation, or it might be a public health project. But talking to the basic scientists as the project is getting developed and looking for opportunities to collaborate. And I have a great recent story from my own work in which I think we had remarkable success in this kind of crosstalk. So I'll talk about that in a moment. Basic scientists can join clinical researchers in the design, follow-up, and interpretation of clinical studies or trials. They can influence the quantity, type, and costs of sample collection. If you collect the right sample, you might open up a laboratory opportunity that if you don't collect the right sample, it's gone. And you got to think about that up front. And subsidies from research grants, from satellite research, indirect cost returns, enlightened trial sponsors, you may be able to get your satellite work funded if it's thought through carefully enough. There may very well be funders. And also, we have to plan learning from negative studies, negative trials, for example. So let's say we do a clinical trial of a new immunotherapy for viral disease, maybe for cancer, whatever it is. What if it doesn't work? Are there experiments that you could be doing that are more functional, that guide us as to why it didn't work? And might a negative study be highly informative if we have the satellite, let's say immunology or virology, that illuminates us as to why that particular antibody didn't work? Yes, the answer is yes. But if you don't have that anchor, all you know is it didn't work. You don't know why it didn't. Next slide. So we've got new platforms, big data platforms, artificial intelligence, natural language modeling, machine learning, and this is transforming the world of integrative structural biology. I mean, the old days you would sort of characterize a protein and you would crystallize it, and you might do screening of 3 million compounds to see if something works. Now we can do lock and key modeling, and we can look at narrowing the screening of 3 million compounds to 3,000 compounds or 300 compounds, because we've got a sense of what we're looking for, because we know structurally what's going on, and then we're modeling using AI what kinds of molecules fit that lock and key structure. And it's really transforming things. You need platforms where we can interact to talk about these, and conscientious university efforts to nurture engagement. Next slide. We're talking to our new Bellini College of Artificial Intelligence, Cybersecurity, and Computing. Now, these are a bunch of computer geniuses that run circles around me in terms of what I'm doing, but the topic areas of the kind of research that I'm doing interest them. They think they can help apply their methods to help me. And that doesn't make them an expert in HIV or an expert in SARS-CoV-2 or an expert in Zika virus or anything else, but they can have me as a translator working with them. And open access to data between networks. We're working on this. Some people don't want to share, but insofar as we can have data that are available, we can get this crosstalk going. Next. And if it's easy to find and understand the trial, the specimens, and the data beyond one's own institution, it can open up real opportunities. I've been very involved with the NIH-sponsored HIV Prevention Trials Network, and we've been very aggressive at making our data and our specimens available. And I can tell you all sorts of stories where investigators who had nothing to do with the HPTN have been able to exploit that for important research. And that's what we're trying to get to, you know, a little less selfishness, a little more vision. Next slide. So now I'm going to give you that story that I was telling you about, virology translation research from a vaccine effectiveness study in the Dominican Republic. Next slide. So I got a call from a colleague in Dominican Republic saying that he, along with the Chinese manufacturer of the inactivated vaccine, wanted to see whether it had actually been effective in real world conditions. And I remember my first reaction was go, inactivated vaccine. Well, that probably wasn't too successful. But I resisted the urge of being a negative, nattering nabob of negativism, and I decided to research it a little bit. And there was a little bit of information out there suggesting perhaps that it wasn't such a bad vaccine for the ancestral and Delta strains. And that opened my eyes a little bit. And I said, okay, I'll try to help you design a study. I didn't know if it would be important in my career or whatever, but I had my friend in the Dominican Republic. I actually knew one of the Chinese investigators, and we forged an alliance. I was at Yale at the time, so it was Yale, and we had some data management experts at Stanford. We had the Chinese vaccine manufacturers, and we had the Dominican clinical scientists and public health scientists, and that was the team. So the more I looked into it, the more interested I was, because the inactivated SARS vaccine, we know it's not as immunologically potent as mRNA, but there were more than 8 billion doses that were given. And some people might say it was better to wait for, it would have been better to wait for a better vaccine. This is waste of money. We shouldn't have done it. Others would say, use what we have available to us. That's what we ought to do. And all of us want to say, were these worthwhile or were these a waste of time? So I got really excited. And then came Dr. Hara's paper in the New England Journal of Medicine, where they did a completely different design study. They did a prospective cohort study nationwide, and they said it worked. It was very effective at reducing severity of disease in the country of Chile. So I said, aha, we may not end up in the New England Journal, but we need to, that's been done, but we need to do this study. This is important. We need to replicate Hara's findings. Next slide. So we did a test negative case control study. So let me tell you what that is. If you meet clinical criteria for possibly having COVID-19, you got the cough, you got the fever, whatever, you have those criteria. Well, you might have COVID-19, you also might have influenza. You might also have RSV. You might also have any number of parent flus and, respiratory viruses. I'm going to exaggerate slightly, but they all present the same way. Yeah, sure, there's some variations on the theme, but the average clinician can't tell by just looking, you have flu, but you have COVID. You can't tell. So the test negatives are people who test positive for COVID-19. And those are the cases, and the controls are the people who test negative. So everybody's sick, but some of them are sick from COVID, and some of them are sick from other diseases. And you can imagine the 2x2 table against vaccinated against COVID versus not, and imagine how that could be informative. This is really real world effectiveness. It's not efficacy that we do under controlled clinical trial conditions. But effectiveness is, how is it actually being deployed in the real world, and is it making a difference in public health? So that is what we did. And we had 10 hospitals in 5 provinces. This is the Dominicans using their entire country, because they said, well, we want to have generalizability. So we want to have hospitals in rural areas. We want it in the north of the country, the south of the country, the far east of the country. And we estimated vaccine effectiveness with the multivariable adjustment for demographic characteristics, which is kind of more what I do for a living in the field of epidemiology. Next slide. So this is where Dominican Republic is, for those of you who don't recall. There's Haiti, there's Puerto Rico, there's Cuba, there's Tampa, right there. So, and these were the regions, the capital city, the region around the far east, Santiago, the second largest, and then the north. So some diversity. Next slide. Now, this was our finding for symptomatic COVID-19 disease. So don't make the mistake so many people make saying this is protection against infection. we're not doing daily, viral load assessments and everything. We don't know anything about protection. Lots of COVID cases are asymptomatic. So this is protection against symptomatic disease. So be precise in your language. Don't say protection against infection, because that would be wrong. We're not assessing infection. And sure enough, with partially or fully vaccinated, fully being 2 doses, partially being 1 dose, we did see modest vaccine effectiveness. And the other big predictor was age. There was a trend towards obesity and comorbidities increasing risk. This was borderline, significant. This was not quite significant, but it made sense from what we knew about COVID. But now the next slide is hospitalization. And hospitalization is actually what we're mostly interested in. It's fine if we reduce your symptomatic disease, but what we want to do with vaccines is prevent you from getting hospitalized, be put on a ventilator, dying. And that's how vaccines work. Vaccines give you the immunologic memory so that when you encounter the real world virus or organism, you have seen it before and you mount the so-called anamnestic response. Amnesia, I don't remember. Anamnesia, I do remember. Sorry for the awkward wording, but that's what we call it, an anamnestic response. So the anamnestic response leads to a much quicker uptake of antibody being produced by the plasma cells, because the memory T cells have seen this before and they send the instructions through the cytokines right away, and the plasma cells mount the immune response. And sure enough, as is often typical for vaccines, this is where we saw 70% vaccine efficacy. And that answered the question. It was not a waste of time for the Dominicans to have used the vaccine they had available to them, a very low cost vaccine. They did not have mRNA vaccine available to them early on. As you remember, the high income countries of the world bought up all the early stuff, and Africa, Latin America, Asia had to get what they could get. Next slide. And here, comorbidities, obesity, and age all were risk factors. So the inactivated SARS-CoV-2 vaccine was highly effective in preventing hospitalization. Also death, but we didn't have enough deaths to have it be very statistically robust. So I'm not highlighting that. But the trend was just as strong for preventing death. And this was in the time of the ancestral and Delta strain, sometimes called Wuhan strain, sometimes called Alpha, different terms. And the DR findings turned out to be not only supportive of the New England Journal article from Chile, but studies in Brazil, in Indonesia, and Turkey all said the same thing. And that's kind of fun for somebody like me. Consistency in clinical research is sometimes hard to come by. And in public health research, it's sometimes hard to come by. There's so many examples where it worked better here than there, but this was rock solid. So the answer is widespread use of vaccination is a major tool for reducing serious disease and death. And sometimes an inferior product is good enough for the purpose at hand. But now that's not actually the story I'm going to tell you. That is the background story. And when Dr. Eddie Perez-Ten, my close collaborator in Dominican Republic, told me a story that the Dominican government had just made a major mRNA purchase from one of the big manufacturers, and they were thinking about boosting people with the mRNA. I said to myself, oh my goodness, we really could do an important study here. And I called Akiko Iwasaki, a very talented Japanese-American scientist at Yale, who's a legend in this field, and also doing important long COVID work. And I called Akiko, and she said, here's what I would recommend. And she kind of told me what I knew she would tell me, is to get the bleed just before the vaccination, and then you know, 1 month and 6 months and this sort of thing. And I didn't know exactly how often she wanted it, but as an immunologist, she knew what experiments she was going to run. And she said, you know, I have some funding that can run these if you can just get the money to ship them. I said, I'll find the money to ship them. Next slide. We still have guests waiting to join. I hope we're letting them in. So keep in mind that this was early on. So that the notion of boosting with heterologous vaccine was still unknown. We weren't quite sure if heterologous vaccination was going to be as good as homologous vaccination. If we know that mRNA is more, gives you a more vigorous immune response, do we really think that the shorter lasting and less broad immune response from the inactivated will be good enough? As maybe we should do, maybe we've got to ignore the inactivated and just start vaccinating as if we're going from scratch. Because by this time we were getting into the Omicron era, and we had different vaccines circulating, which required a broader immune response from the vaccinations. So of course, you know, we've had this evolution of all of these different variants, which varied from transmissibility, severity, vaccine efficacy. So we were in a moving target. Next slide. So when you look at this spike protein, you may remember the work of McClellan and Graham and others, where this sort of trimer sort of opens up or closes. And it was that discovery, that fundamental virologic discovery, that enabled them to figure out what kind of vaccine was going to be used. Because if you used it against the wrong structural formation, you were going to miss the boat. So, you know, fundamental discoveries at the laboratory level that made huge differences in vaccine design and deployment. Next slide. And this is what the fusion protein looks like, and terminal, the binding site for ACE2 receptors, the anchor. Next slide. And you have this ACE2 binding site represented sort of in pink, and the furin cleavage site sort of in red. And this is the kind of designer, you know, structural virology that vaccinologists now can use in a very concrete way to help design vaccines. It's no longer guesswork. It's no longer empirical. It can be designed. Next slide. And these were the variants of concerns that you recall emerging in the UK and South Africa and Brazil and India. And then November 2021, the South Africans reported, next slide, the Omicron, which kind of changed the landscape, because Omicron wasn't as susceptible to neutralization from the inactivated vaccine. So that opened up the whole question that we were posing. Next slide. So we asked the question, what is the effect of mRNA vaccine boosting in persons who've received the 2 doses of inactivated vaccine? So if you got 2 doses of inactivated vaccine, you were eligible for our study if you were coming in wanting your mRNA booster. And we collected the blood specimens, as I mentioned, and this we felt was vital information for all nations that had been using inactivated vaccine, which was like 50, 60 countries around the world. I made up that number, but it was at least that number. Next slide. Whenever you make up something, you have to admit it. So this is what came out from Carolyn Lucas and Akiko Iwasaki and colleagues. This is what we found. So previous to the booster shot, if you got a booster with the inactivated vaccine, didn't do anything for you with Omicron, but you still had pretty good response to Delta and Ancestral, which is what our public health study had suggested. Next slide. And we're using, you know, the plaque neutralization, reduction neutralization test. And 28 days post booster with CoronaVac and with Pfizer, we were getting much more robust responses. You know, essentially what we felt were going to be protective responses. Next slide. And then we asked another question. And by the way, that, you know, I remember when I saw that and I said, you know, Akiko, this is hot stuff. This is highly relevant for policymakers around the world. If she wanted to publish it in Virology, I said, I think this is Nature Medicine stuff. We need doctors, we need public health people, we need a broader array of people to see this. And I think Nature Medicine will get it right away. And she goes, are you sure? You know, really? Because, you know, it's hard to get into Nature Medicine. I said, I usually can't predict what journals to get into, but this I think we can get. And it was reviewed favorably and accepted in record time. And it's been cited, I don't know, 400 times. It just published a couple of years ago. So it really was important, I think, for the field. We had another opportunity. Does homologous versus heterologous in those fully vaccinated with the CoronaVac, the inactivated product, do they induce distinct humoral responses? Is there any age group difference? So we found that IgG against Omicron were substantially reduced in participants receiving the homologous versus heterologous, but this was the novelty here. It was especially dramatic in older persons, especially dramatic. So that took a whole nother analytic framework to get an answer to this. It took months after the first paper was published before we were finished with this. And it confirmed our original findings, but then added this age group difference. Next slide. And this one was also, boy, they're slow today, these slides. This was also in a major journal, Science Translational Medicine. You know, you could be boosted either with the Pfizer. A few people were boosted with the Oxford vaccine, and some were boosted with another CoronaVac. And you could see anti-spike boosting being a little more robust with the mRNA compared to the other two, and you could see real failures in the booster with the inactivated vaccine. But the next slide is really the interesting thing.
I can see it, and it's great.
So take a look at the age factor. If you're 20 years old, even boosting with the inactivated CoronaVac wasn't too bad. Take a look at people my age. You were getting nothing for your money here. And that was exceedingly interesting, that there was this age-related factor that we need to learn about, we need to understand. But it was immediate guidance that don't bother vaccinating the most vulnerable people, the older people, with a third inactivated vaccine, because it ain't gonna work. Next slide. And this is just the kind of data. I don't have time to really walk us through because I'm looking at my clock and I'm almost done. Next slide. Let's just go to the note. And this is the kind of data that we were doing by age group. Homologous induced lower responses, and especially lower in the elderly. It was vaccine specific. It wasn't lower with the Oxford or the mRNA products. You saw that age didn't matter. So it was specific to the inactivated. And the homologous boosters did not improve neutralization response against Omicron for older persons. Next slide. And I don't have time to talk about it, but there was yet a third opportunity here, led by folks around the world. This was adding our data to data from Brazil, data from Asia. Kiko is connected all around the world, and she pulled this together. And there was material in the literature saying that fetal defects might be caused by mRNA. These were studies of 6 people. 8 people. They were just not epidemiologically sound studies. And also that anti-syncytin 1 antibody production, which is not a good thing, was stimulated by this. And we produced what I think is the definitive paper. And we played a minor role, the Dominican study, because we conglomerated data from all over the world, published in PLOS Biology, which I think was important. Now, you see what's going to happen is certain people, to be remain unnamed, RK Jr., will take this and find that 8-person study and ignore the definitive study. So that's how data get manipulated. Next slide. And I got nothing against studies of 8 people as long as you understand that their limitations and understand those are hypothesis generating. They're not testing hypotheses. So finally, and I'm almost done, and I hope you get how a public health project that is launched by the prepared mind with the kinds of basic virology, immunology connections can be far greater than what you had intended. And the only regret is I didn't have Akiko in the room when we actually designed the original study. Turned out it didn't matter much, but it might have. And that would have been a missed opportunity on my part. So we have a lot of need for pandemic preparedness, epidemics, pandemics, and of course, pandemic flu is our greatest threat. Epidemics, serious outbreaks in restricted communities or regions, pandemics that jump continents, and then pandemic flu. We sit in dread of genetic reassortment when I have the human virus and I also go to the farm and I get the H5N1 and I have a reassortment so that the backbone of a human adapted influenza virus now has a novel sort of antigenic challenge that my body has never seen before, and I get very, very sick. And then I transmit it to you and you and you, and we have the shift as opposed to drift. You know, this season we have a drift. We have the subclade K, which is now dominant in Japan, dominant in UK, and looks like it's going to be dominant here as well. It emerged from the Southern Hemisphere. We don't hold you personally responsible. But every year, either something emerges from the Northern Hemisphere and gets transmitted to the Southern Hemisphere, because we have opposite seasons, right? Or the other way around. It emerges from the Southern Hemisphere and we get to inherit it in the Northern Hemisphere. So, and in tropical countries, you've got year-round influenza. It's not season. You can have these things emerge. So the bad news is that this year's vaccine is not going to be as good as others because it's a brand new subclade. So we didn't develop the vaccine 9-10 months ago for this subclade. But the good news is it looks like it's still going to be very solid for preventing serious disease. So we definitely want people to get vaccinated. Next slide. And we all know about the Asiatic flu of the 2 centuries ago, the so-called Spanish flu that we talked about this morning from 1918, Asian flu and Hong Kong flu. So we've had pandemics in 1889, 1918, 1957, 1968, less dramatic in the 21st century, but we've had one pandemic at least. And of course, COVID, which is a different virus. But you see the estimated deaths, and COVID is more in this category in terms of the Spanish flu category than any of the others. Next slide. So we mentioned that modern travel is such that 95% of humanity can visit each other within 48 hours. We have much more densely populated urban areas now. We have the great megacities of the world. We used to think that New York and London and Paris were big. Now we think that Karachi and Sao Paulo and Lagos, I mean, much, much bigger. Shanghai, much bigger cities out there. In 1918, we had civilian and military overcrowding. We withheld public information, and the global population was approximately 1.8 billion. Now, I think you all know that the global population today is over 8 billion. And crowding is nothing like it was back then. You know, now we have real crowding. Now we have dense urban centers. Next slide. And I'm going to skip this slide because I just said it. Next slide, because I'm out of time. And the next pandemic, is it going to be influenza? Is it going to be another coronavirus? Is it going to be disease X? Have you heard that term? The WHO is using that a lot, disease X, which is basically saying, who knows what's out there. And WHO correctly predicts that we're going to have periodic influenza and other pandemics. That's a certainty. That's a certainty. We know that there will be viral shift in flu at some point. It's unknown when it will occur or how severe it will be. We don't know actually the organism. And it is in the context of declining immunization rates. So this anti-vax phenomenon that seems to have taken control of certain elements of social media, and well-meaning parents don't want to vaccinate their kids. Why? Because they've been misled. There was a gentleman, a Mennonite farmer in West Texas, whose daughter, 8 years old, died of measles. The anti-vax people got a hold of him and convinced him that since the daughter died of pneumonia, the doctors were doing the wrong thing and she got pneumonia, not realizing that that's why kids die of measles, because of pneumonia, pneumonia, and secondary infection. And he's now on the trail of the anti-vax folks, trying to talk people out of vaccinating. His daughter, age 8, died of measles. So we've got our work cut out for us to re-engage the general public. And I think Maggie's paper here is worth reading. This is a nice paper on modeling reemergence of vaccine eliminated infectious diseases under declining vaccination in the US. And my paper in JAMA, which is just 1,000 words, we probably should give, yeah, it's 1,000 words. It'll take you 8 minutes to read. And I tried to make an argument as to why leaders in Florida, where we're sitting now, recommending getting rid of mandatory vaccination is not a good idea. And I tried to write it in such a way that Parents and Teachers Association could share it, doctors could share it with patients, try to get a counter argument going. Next slide. So that is it. Keep in mind that GVN is keen to collaborate and assist its international partners in translational and implementation science research, not just basic virology. I love basic virology. That's why I agreed to take this job in the GVN, even though I'm not a virologist, but I hope that we can do more than that. We can accelerate development of products through translation. So thanks for your attention, and happy to take questions from those of you online. We can probably get rid of this slide if people have gotten my email addresses, and happy to communicate with any. By the way, happy to share these slides. Anybody who wants these slides online, or just send me a note and I'm happy to send them out.
Thank you, everyone. Thank you, Dr. Vermund. That was a fascinating talk. I encourage anyone online to put their questions in the Q&A, and I will open it up if there are any questions in the room.
Thank
Yes,
you.
please.
I very much enjoyed that. I just couldn't move past asking this question about the Dominican Republic study. At that time, in those that were test negative, was there any baseline antibody testing to exclude that they hadn't had previous infection? Because that would have surely been a confounder in looking at the results and later what we knew about immunity walls. And then with the emergence of the Delta strains, were the diagnostic assays also not a question? Might they have missed?
The second question is easier than the first. We used standard license products that were the same being used in the US, UK, etc. We didn't do any better than standard diagnostics, but we certainly didn't do any worse. We did state-of-the-art diagnostics. The question as to whether the test negative group might have been infected with COVID at a higher rate than the test positive group is a very legitimate criticism. That's why papers like that don't end up typically in Science, Nature, Lancet, New England Journal, because that objection we can't resolve. We did not have any resources to do that kind of testing at the front end. But you would then have to say that somehow you're in some ways you're more likely to get non-COVID respiratory virus than COVID respiratory virus, to postulate that the test negative group somehow was more likely to have gotten prior COVID and somehow protected against COVID. They had a respiratory virus and they were sick from it. So I think it's a very legitimate methodologic criticism, but I would argue it's improbable. And that's why I got so excited when the Brazilians, the Indonesians, the Turks, all found the same thing that the Dominicans and the Chileans had found. So, and there were different designs. In Chile, it was national cohort. So I think we have good enough evidence as best we can usually manage with this kind of circumstance. But I also think that in the ideal world, with ideal funding and the like, getting that specimen right up front and taking a look at prior infection in that other group. But it would have been tough to do that in the COVID group because they might have tested, positive, and it would have been tough. It would have been tough to do it logistically. We thought about doing it, and we just didn't know how we were going to interpret the results if we ended up doing that, because we knew we had the test positive group, and then we can't ask the question, how probable is it that they had prior COVID in that group? They're going to test positive. Anyway, good question.
All right, Sten, we have a question online. How is vaccine hesitancy addressed across the studies mentioned? Was there any case of viral co-infections with another virus under which the trial performed could potentially have altered the vaccine efficacy?
So I presume that's referring to the Dominican study.
Mm-hmm.
So Dominican Republic has not been plagued by vaccine hesitancy. The cultural norm in the Dominican Republic is that they view vaccines as a plus. They have surprisingly high, for a not very prosperous country, surprisingly high vaccine coverage rates for MMR, DPT, et cetera. And there was very high uptake of COVID vaccines. So we did not encounter a major confounder based on people who turned down the vaccine. Now, plenty of people didn't get the vaccine, which is why we had a study. But that tended to be more vaccine availability. It was, and there must have been some vaccine hesitant people in that group. I won't say there weren't, but we don't think on the biological side it will alter our interpretation of the study. The second half of it.
Co-infections. And
yeah, we don't have, we have now the era of the multiplex assays that can test for 20 different respiratory viruses all at the same time. They cost a lot of money, and we didn't have a lot of money. So we didn't have that luxury. So we more or less had COVID, non-COVID, which you could argue is a little bit primitive and suboptimal. And I accept that criticism too.
I think that's all the time we have for questions. I do have a comment that I'd like to read because I think it's appropriate, from Franco. Excellent presentation, Sten. This is a real example of research. The solution comes before the question. Serendipity is the key of real science of unknown. And I'd like to thank you all for joining our GVN Forefront of Virology webinar series. Dr. Sten Vermund for the fantastic presentation, and we look forward to seeing you here next month.